Good afternoon. I'm Tanmayee. I'm a postdoc at the University of Dundee. I'll be presenting my PhD work on learning shared chromatin landscapes and joint denoising of histone modification assays. I'll start with some background. Inside the nucleus, the DNA is wrapped around histone octamers to form a unit called the nucleosome. These nucleosomes are folded to form tertiary structures called chromatin, and these chromatin fibers form chromosomes. Chromatin accessibility is not uniform across the genome. Heterochromatin is tightly packed, and euchromatin is loosely packed. And this chromatin accessibility is cell type specific. Each unit of the histone octamer has a long tail that is covalently modified with chemical groups called histone modifications. There are over 100 known histone modifications that regulate chromatin accessibility and aid in the recruitment of transcription factors. And these histone modification patterns are cell type specific. Further, these patterns are highly correlated, as you can see in the figure here. Each row shows the enrichment of a histone modification at a particular genomic region in yeast. The correlation happens because histone writers contain reader domains that can read other histone marks. This allows for the reinforcement of modifications through feedback loops and inhibition by other histone marks. In 2019, the ENCODE Imputation Challenge was organized to encourage the use of computational methods to impute genome-wide enrichment for missing cell type assay combinations. Here on the right is a figure of the data matrix for the challenge, with histone modifications on the y-axis and different cell types on the x-axis. Data is only available for squares that are colored. The computational task is to use existing data to impute missing values. Other members of our lab developed EDICE, a new method to solve this problem. This has been published in 2023 Nature Communications. On the right, I'm showing an example of observed histone modification enrichment in blue with the predicted imputation overlaid in red. However, the competition organizers face challenges with evaluation of submitted models. In their paper, they say that this is because large consortia data is collected over many years, leading to changes in experimental procedures and data processing. And this creates a distributional shift. In this work, we looked into this aspect in more detail. Before we look at histone modification data, I'll give you an overview of how ChIP-seq works. ChIP-seq is the most popular experimental technique for measuring histone modification enrichment. Here, I am showing DNA with histone modifications in red. In the first step, the DNA is fragmented. This is affected by the chromatin organization. Loosely packed DNA is fragmented easily, while tightly packed DNA is harder to fragment. For the treatment sample or the ChIP sample, an antibody is added that binds to the histone modification. For the control sample, also called the input sample, there is no antibody added. Both ChIP and input samples undergo DNA purification, amplification, and finally sequencing. Lastly, the respective reads are mapped to the reference genome. The input and ChIP samples have a high correlation for a majority of genomic loci, and this can be clearly seen in the data. Here I am showing 2D histograms of just treatment and control coverage for transverse colon tissue of individual 51F from the ENCODE NTEX dataset. And here I'm showing data for 5 different histone modifications. The x-axis shows average control coverage and y-axis shows histone modification coverage. The presence on the diagonal shows this strong correlation. For the data analysis of ChIP-seq, a genome-wide enrichment signal is computed. To do this, coverage is initially computed for both treatment and control. And then for each genomic location, control and treatment coverages are compared and quantified with a statistical test. This results in a genome-wide signal in minus log p-value, and this minus log p-value is the enrichment that is predicted by imputation models. However, there are some biases from the measurement and analysis processes. For example, in most experiments, control and chip have comparable library sizes, and this results in low coverage for control samples, with sometimes up to 16% of the genome not covered at all. Further, MAX2, the ENCODE recommended tool for ChIP-seq analysis, uses the Poisson significance test. As you already know, the Poisson distribution is asymmetric, especially for lower values of the mean. These two factors taken together results in histone modifications associated with closed chromatin having a lower maximum enrichment on average compared to those histone modifications associated with open chromatin. Also, in a lot of pipelines, replicates are merged, losing variability information. For a given cell type, each modification is analyzed separately. And our question is, can we do something better than this? We propose a new tool called Decoden. It consists of two main steps, deconvolution and denoising. In the next few slides, I will briefly explain how it works. For each genomic bin, the control replicate is written as an element-wise product of 4 terms. DHR is a scaling factor to account for sequencing depth. AB is a cell type specific bias, which includes bias from local chromatin accessibility. MB is the sequence bias, including bias from mappability. And NB is a non-specific signal for other biases. For treatment replicates, we have a few additional terms. KHR is a histone modification specific mixing parameter to model antibody specificity. SHB is the true hidden histone modification enrichment, and this is what we want to estimate using the method. The first step is deconvolution, where we use matrix factorization to decompose a matrix V into two matrices W and K with non-negative entries. In Decoden, we concatenate all samples. So first we concatenate control samples followed by the treatment samples for every experimental condition. And after factorization, we get the matrix W with X, which models the chromatin bias. And for each histone modification, we get a corresponding Y signal. And in the end, we also have a mixing matrix K. The next step is denoising with half-sibling regression. This method is inspired from causal inference to remove the effect of confounders. In our case, the confounders are accessibility and mappability. Here, Y1, X, and Y2 are from the previous NMF step. The true modification signals S1 and S2 are unknown. This is what we want to extract. Accessibility and mappability are also unknown. To estimate the histone modification signal, we subtract expectation of the modification signal given chromatin bias from the modification signal. And this regression is done in log scale for numerical stability. To summarize, in the deconvolution step, all replicas are concatenated into a big matrix that is factorized. We estimate the chromatin bias and histone modification specific signal. For example, here, the purple replicate is decomposed into a blue chromatin bias and red histone modification specific signal. Next, in the denoising step, half-sibling regression is used to extract the true enrichment pattern by minimizing the bias introduced by accessibility and mappability. Here are the results before and after Decoden. As you can see, the diagonal that we saw earlier in measurement data is completely gone. So we can say that Decoden minimizes the chromatin bias in histone modification assays. And this is an example from a genomic locus. This is data from a deeply sequenced data set for lung adenocarcinoma cell line. Red shows H3K4 3-methylation, and blue shows H3K27 3-methylation. The true peaks on either end for H3K4 3-methylation are preserved, while the sequencing bias in the middle is removed. When we look at genome-wide correlations, the treatment replicates are reasonably correlated with control replicates. The Decoden estimated signal simultaneously has low correlation with control samples and higher correlation with treatment samples, which again indicates that it minimizes the effect of confounders. The mixing matrix from the matrix factorization step provides a way to qualitatively assess antibody specificity. Here I am showing the mixing matrix for two different antibodies which were independently assessed by the chromatin antibodies database. The antibody with higher specificity, which is the first one, has a higher mixing parameter for the ChIP-seq signal compared to the chromatin bias. Now let us look at individual epigenomes from the NTEX dataset. Here I'm showing two examples of genomic loci in transverse colon where a HTK4/3 methylation peak is more prominent in 37M, shown in orange, compared to 51F, shown in green. The histogram below shows the average coverage over different modifications in the highlighted genomic region. As you can see, replicates for individual 51F have higher variance, leading Decoden to estimate a lower enrichment value. Here I'm again showing you data for 51F from NTEX for transverse colon. You may have noticed already that not all measurements are equally good. For example, for HCK4/3 methylation and HCK27 acetylation, there are several genomic locations with high treatment and low control coverage, indicating good data quality. For the rest, which are HCK36/3 methylation, HCK27/3 methylation, and HCK4 monomethylation, the data quality is not as good. This is also reflected in the Decoden estimated enrichment. When we do a genome-wide comparison across individuals, we see that the chromatin bias has the best correlation with a value of 0.52. Surprisingly, histone modifications that have a better signal, such as HTK4/3 methylation and HTK27/3 methylation, show a wider range of values between individuals. To conclude, what I showed in this talk is that ChIP-seq data is confounded by biases from accessibility and mappability. We introduced a tool called Decoden that jointly analyzes measurements from different histone modifications from a fixed cell type. It has two steps: deconvolution with matrix factorization and denoising with half-sibling regression. It minimizes the effect of confounders and can qualitatively assess antibody specificity. We applied Decoden to the NTEX dataset, which profiles individual epigenomes. However, data quality is a major bottleneck. Decoden is available on GitHub. It is open source, so if you have histone modification data for a fixed cell type for different histone modifications, I welcome you to try it. And the preprint is on bioRxiv if you want to know more details. I also would like to add that I'm looking for postdoc positions. Thank you for your attention, and now I'll take questions.
Thank you for the presentation, and questions are available now. So if you want to have a question, there's 4 mics. So in terms of data quality that you were talking about, what could be done to improve the data quality? I guess it's from the experimental side where the problems are.
Yes, I think for most experimental designs, they don't spend enough money on the control experiment, which maybe makes sense. But if we don't profile the accessibility and chromatin bias, we can't really know if it's a true enrichment or is it just from sequencing. And these enrichments are now commonly used in a lot of other models like imputation or prediction of ChIP-seq signal and so on. So in a sense, we are only predicting noise and not really signal. So there's maybe, I would say we need to push for better quality data.
So in terms of quality, you mean here the lack of controls?
The lack of, like the library sizes for control is too small. So I didn't look at the entire consortium data, but up to 16% is not covered at all. So that's some significant part of the genome that we don't know.
There's not enough depth.
Yes,
in the
exactly.
Okay, got it. All right, thank you. Any other questions? There's one there, I believe, number 1. Yes, thank you.
Thank you very much for the talk. And I was wondering, because you said that you compared it with Max 2, but have you compared it with Sizer 2, for example, that takes into account replicates? Or your tool?
Sorry, did you say Sizer?
Yes, Sizer 2.
Yes, we have compared it to other tools. There's a comparison with simulated data in the supplement of the paper. And we found that it performs on par. So also I would like to say a lot of the other methods focus on peak calling, which was not our emphasis here. So I hope that answered your question.
Okay. Thank you. I will take a look at the paper.
Thank you.
Any more questions? I don't know if there were questions online. Sorry, I cannot connect, but.
No questions online.
Okay, very much. Well, thank you very much again to the speaker.